Welcome back. So, up to now, our story has been earthbound chemistry, geology, early Earth, primordial soup. By the 20th century, does something breathtaking. Humans leave the planet. For the first time in 4 billion years, Earth's biosphere sends a representative into space. A tiny primate with a plastic helmet. And with that, the search for life becomes cosmic. This is a lecture where astrobiology becomes a field, not an idea, philosophy, dream. The years 1950 and 2000 transform everything. The rockets, spacecraft, telescopes, molecular biology, life detection experiment, ice moons, interstellar organic SETI Institute. So lots. And come on, let's begin. Modern abiogenesis era. So, before diving into space missions, let's briefly examine the revolution happening in the chemistry at the time. By the late 20th century, researchers realized something profound. Life is not a molecule, life is a system. So, modern prebiotic chemistry shifts toward autocatalytic networks, reactions that amplify themselves, like early metabolic seeds and chemical selection, primitive survival of the fittest among chemical pathways, coacervates and peptide droplets, protocell like compartments forming naturally, and protocell evolution, droplets that grow, divide, fuse, and compete with each other, and prebiotic RNA and peptide pathways. Chemistry gets closer and closer to biochemistry. This system's chemistry view brings together lipids, peptides, RNA, mineral networks, environmental cycles, all interacting as proto-life. This is the intellectual backdrop as NASA begins actual astrobiology missions. So this era is really crucial. By mid-century, humanity had all the pieces. There were naturalistic explanations from Darwin, Pasteur, Operon, and lab synthesis of building blocks like Miller and Urey experiments, understanding of DNA, RNA, and proteins, planetary science, spacecraft. This is when astrobiology becomes something testable. The 20th century created the framework for every mission, every experiment, and every search strategy we use today. So let's go with one of the influential figures, Carl Sagan. Sorry. No single figure shaped modern astrobiology more like Carl Sagan. He was a scientist, visionary poet, communicator, and yes, a romantic about life in the universe. His contributions were enormous. Titan's atmospheric chemistry, he simulated Titan's atmosphere in the lab and produced orange organic haze toluens. He also worked on organic molecules in interstellar space. He helped us show complex chemistry is widespread. Ultraviolet photochemistry, Sagan demonstrated that ultraviolet light drives essential prebiotic reactions. And also Europa ocean speculations. He was one of the first to suggest Europa's subsurface ocean could host life. And public advocacy. Through Cosmos, he turned millions of people, like myself, towards science. Carl Sagan didn't just do science, he made science beautiful, poetic, and reachable. He also co designed the Voyager Golden Record, Earth's message to the stars, and actually included a kiss between two humans because, in his words, love is not irrelevant to our species. Very, very romantic. So, in 1959, something historic happened. NASA creates the Exobiology program, the first official government funded scientific program devoted to prebiotic chemistry, origin of metabolism, protocells, membranes, RNA world, microbial survivability, planetary habitability. This program created the field we now call astrobiology. Before 1959, searching for life was philosophy or fiction. After 1959, it became government funded experimental science. When NASA was founded in 1958, some scientists actually joked, "We are sending rockets to look for little green men," they called it. By 1960s, they realized the subject was much deeper and far more difficult than they could imagine. And there were a couple important missions at the time, like Mariner and Venera. So, Mariner and Venera, the space age begins with excitement and also disappointment. So, Mariner 4 in 1965 was on Mars and first close up photos it took. So, scientists expected rivers, vegetations, maybe even ruins of civilization. Instead, they got a barren, cratered desert, no canals, no ocean, thin atmosphere, freezing temperatures, and Mars goes from perhaps alive to probably dead. And in the Venera missions, the Soviets land multiple probes on Venus. The results were 460 degrees, 92 atmospheres of pressure, sulfuric acid clouds, crashing greenhouse effect. Venus became the solar system's warning sign. These missions created the dead solar system model that dominated 70s. It was a scientific heartbreak, but a necessary one. Before continue, I need to talk about the space race. Soviet era, the early space missions, the Cold War scene. So let's zoom out to something crucial. The Cold War didn't just shape politics, it shaped how we think about life in the universe as well. When we think about astrobiology today with its missions, lab, telescope, so much of that infrastructure came from geopolitical competition between two superpowers. So the Soviets were first to space, the first biological experiments. The Soviet Union kicked off everything in 1957 with Sputnik 1, that little probe you see here, the first artificial satellite, and people panic. The newspaper thinking the Soviets could now drop bombs from space. In reality, it was a metal ball that simply beeped. And then they got Laika in 1957, the first animal in orbit. A stray dog from Moscow streets becomes the first living Earth organism to leave the planet. And here's the interesting part: the Laika's flight was considered a biological experiment, how the organisms react to space. And Soviet scientists later said, "We did not learn enough to justify the loss of life, but Laika set the foundation for human spaceflight biology." And there were lunar program in 1959 and 66, first probe to reach the moon, first images of the far side, first soft landing. These missions forced scientists to ask, could microbial life survive lunar conditions? Spoiler, no, but that question triggered the development of planetary protection protocols we're gonna talk about later. So Cold War is explosion of funding for life sciences. This is one of the most important points for astrobiology. Government realized space was not just about rockets, it was about biology, psychology, geology, engineering, chemistry. Every field was suddenly part of the grand mission. So NASA Exobiology program created in 59 was literally born because the Soviets scared the US. The US wanted to understand, could life exist on Mars? If yes, could the Soviet find it first? And what if they brought microbes back? This fear accelerated astrobiology by decades. So actually, Soviets sent tortoises to the moon, Zone 5 mission in 1960s, and they become the first Earth organism to circle the moon and return alive. And the scientists joke, technically, tortoises reached the moon before Americans did. So it was a really harsh race. And for NASA, there were Apollo and lunar science and global impact on astrobiology. So while the Soviets drove the early years, the Americans delivered the spectacle. In 61 to 72, there was the Apollo program and the moon landing. So Apollo was not just a national pride, it was a scientific revolution. The Apollo astronauts were essentially geologists, field biologists, photographers, lab workers all in one. They brought back 380 kilograms of moon rocks. These rocks rewrote our understanding of planetary formation and volcanic history, early solar system bombardment, and water distribution in early Earth Moon system. So NASA trained the astronaut in geology in the desert of Arizona, and some astronauts says we felt like space geologists, but we loved it. So we will talk about the planetary protection later. But Apollo missions were so worried about moon germs that astronauts were quarantined for 21 days and their suit sample tools were isolated. The lunar receiving lab was built like BSL 4 biohazard facility. Imagine Buzz Aldrin, the second man on the moon, sitting in a quarantine room saying, I just been to moon, now I can't leave my house. This paranoia led to modern planetary protection rules actually still used by Mars missions, Europa Clipper, James Webb contamination protocols. But to cover this all up, the Cold War creates a new scientific field, period-shaped astrobiology through funding, competition, planetary science revolutions, and public imagination. We told that, and after the Mariner and Venera missions, there was a renewed optimism. So, after decades of pessimism, new missions revealed something astounding. Valley networks, ancient river systems carved by flowing water, the Jezero Delta on Mars, a river delta identical to Earth's, layered sediments, deposits formed in long lived lakes, and suddenly Mars wasn't dead, it was ancient, wet, and possibly habitable. Modern Mars is a desert, ancient Mars was blue. This transformation, astrobiology from hopeless to hopeful again. And let's talk about the one of the important missions, the Viking mission to Mars. In 1976, the United States landed two spacecraft on Mars, Viking 1 and Viking 2. These remain the only dedicated life detection experiments ever sent to another planet. Vikings performed labeled release experiments, gas exchange tests, pyrolysis and GCMS, and soil metabolism assays. The results organics not detected. We now know perchlorates destroyed them during heating. So positive results, but chemical, not biological. So to conclude, it was ambitious. The scientific community argued for decades, and some argue still. But Viking changed everything, forced astrobiology to become more rigorous, led to modern biosignature science that we will discuss later, inspired future missions like Curiosity and Perseverance. Viking was a turning point, frustrating, brilliant, historic, but very important. So, actually, Gil Levin, one of Viking's experiment leads, insisted until his death, until his death, that Viking found microbial life. Most scientists disagree, but his persistence became part of the mission's legacy. And continue with another important development. The SETI begins. Humanity calls out to the stars. So in 1974, humanity sends its first intentional message to another world, what we call the Arecibo message. We will discuss shortly here. This was the message we sent, and it so displays the DNA structure, a human figure, our solar system, excuse me, our solar system. So Arecibo telescope schematics is given there, and population data, elements needed for life. It wasn't meant to actually reach aliens. The cluster will have moved by the time the signal arrives. It was symbolic. The SETI, the search for extraterrestrial intelligence, had begun. For the first time, astrobiology isn't just about finding life, but also being found as well. But also, the sending message to space has very limitations today because of many concerns that we might discuss later in our series. Actually, if you know the movie Contact, it was influenced by the SETI project, and also the present of the satire. But let's continue with our lecture. The molecular origin of life theory is rising as well. So, from the 80s onward, biology explodes into a molecular revolution. Ribozymes were discovered 82, 89, RNA world revived. There's a proto-metabolism models, membrane first hypothesis, fatty acid vesicle, peptide RNA coevolution, autocatalytic state, genetic innovations. So molecular biology gives us tools to study life as a smallest scale. And imagine its beginning at the largest. And at the same time, the astrobiology revolution as well. So from 2000 to present, this is where modern astrobiology truly blossoms. Mars Science Laboratory, Curiosity, riverbeds, conglomerates, clay minerals, organic molecules. Mars proves it was once habitable for millions of years, and this changes the equation. Life might not be possible. It might had, have at time. And also the ice worlds has been introduced. So this is a moment it expands far beyond Mars as well. We are talking about the Europa, Enceladus, and Titan, like Jupiter's moons, and their findings. So Europa, there was a Galileo mission, and we find out it seems like a global subsurface ocean and cracked ice surface and tidal heating, possible hydrothermal vents underneath where we uh, imagine origin of life took place, and there's a potential for a second origin of life happening in Europa or already happened, and there could be life under those water. And Carl Sagan would have loved all these. And there's a Cassini missions to Enceladus, which was really interesting because biggest surprise because they find out in the real images. If you search them online, you can see these water plums into space from the volcanic activity, and this organic there could be organic molecules, salt, silicate particles, evidence for hydrothermal activity, and Celadus became the most promising world outside of Earth for life, and still I would argue it has the most potential for life. And there was Titan, and there was Cassini and hydrogen missions, and there were methane rivers and lakes and rich organic chemistry, atmospheric photochemistry, prebiotic conditions unlike anything on Earth. And now NASA's Dragonfly mission will fly a rotorcraft across Titan's dunes and lakes. It's really interesting. I remember there was a idea, a speculation about the methane production, and if an organism is producing it, and one of the missions there should be leave oxygens and follow it through turning into methane, and thinking might be a bacterial origin behind this metabolism in Titan. So there could be life. Um, so the organic haze on Titan is so thick, actually, that if you stood there, the sun would look like a dim orange glow, as if you were inside a global smoke chamber of prebiotic chemistry. So these all are really important and matters. This era from 50s to now tells us that astrobiology is not a fringe idea. It's a rigorous, multidisciplinary scientific program supported by chemistry, physics, geology, planetary science, molecular biology, spacecraft engineering, philosophy, and information theory. We gone from speculation to experiments to now missions to entire worlds of possibility. So modern astrobiology stands on the shoulders of pioneers we met today. So, in the next lecture, we enter the age of exoplanet discovery, biosignatures, and the question that began our entire course, are we alone in the universe? So, thank you, and I'll see you in lecture 7.